Hey y'all, this is Dr. Fodry. We're going to do section 1.3 of chapter 1 of your Bio 111 course. Just so you know, I'm recording this lecture at home, so you may hear a couple of barks from my dogs, Peaches and Maggie. I apologize in advance. They are very enthusiastic about biology, as am I. So let's get started. Okay, so this third section of your chapter 1 of Bio 111 textbook is on the scientific method and how the scientific method is used to make new discoveries in science. I kind of love this part of the book. This is why, because scientific method is at the heart of how scientists discover things about the natural world. And I love the fact that even the word science from the Latin means to know. It comes from the Latin scire, to know. And so I think that really well expresses the purpose of science, and that's to figure out new things about the natural world. So we're going to do this scientific method. It's going to be a process of inquiry where we search for information and explanations to learn more about the world around us. The way that we're going to do that, well, we're going to do some things that may have familiar names or terms to you. The scientific process, the scientific method, we're going to do things like make observations and form hypotheses and test them through experiment. So you may have seen this before, but I wanted to go ahead and talk about it because creating an experiment, creating an experimental design and using the scientific method is going to be part of every single test. It's going to be kind of a problem called a problem-solving problem on each of your Bio 111 tests this semester. So I wanted to make sure I give you guys the format for how to answer those problem-solving questions for your tests so that you're prepared. Okay, first step in the scientific method is to make observations and to define a problem. We do this because as we're looking around at the world around us, oftentimes, as a scientist, as a biologist, we'll notice something about the world around us that prompts us to ask a question. So observations and facts, those things are called data, and data are just, systematic, organized observations about the world around us. There are two types of data. They are qualitative. That qualitative data is kind of like a description, data that's recorded through the use of words and noting the characteristics of substances or individuals or animals or organisms around us. Quantitative data is the second kind of data, and quantitative data are numerical measurements. So these are type of data that have numbers attached to them. Things like blood pressure, maybe things like the number of leaves on a plant. Maybe numerical data would be the amount of time it takes for seeds to germinate. Any kind of data that has a number attached to it would be considered quantitative data. And usually we can express these quantitative data in the form of charts or graphs or tables. Okay. The other thing that I need to generally talk about is two different types of reasoning, inductive reasoning and deductive reasoning. These two types of reasoning are logical processes by which we kind of navigate the scientific method. So once we make that observation and we notice things about the world around us, we start doing things like asking questions and creating hypotheses and generating testable predictions and making experiments. And the way that we do that is with logic and reason. And two different types of reason are inductive reasoning and deductive reasoning. Inductive reasoning draws conclusions through the logical process of induction, and that just means that this process of reasoning goes from lots of specific observations to generalizations. So, in other words, we can have lots of different examples of something that we notice in the environment. Maybe I notice that many of the trees that I look at have leaves, or many of the trees that I look at their leaves are green, or many of the trees that I look at, they have a vascular tissue system that allows them to transport water and nutrients from root to leaf. That's kind of the beginning point, is lots of specific observations, and that's where we begin for inductive reasoning. Where we end up is a generalization, like a big picture idea. So out of the many different observations that many of the trees that I see have leaves, I would make the generalization, or I would use inductive reasoning to state that in general, trees have leaves. If we are talking about the vascular system of trees, maybe again, I would make many specific observations of trees having vascular systems, vascular tissue, xylem and phloem. And again, through inductive reasoning, right, I would come up from those lots of specific observations with a general big picture idea. In general, trees will have vascular tissue, xylem and phloem, that allow them to transport water and nutrients from root to leaf. This is kind of a bottom-up approach where we take lots of different specific instances and facts, and we kind of build those together to come up with a broad umbrella idea or concept or generalization. Okay. The second type of reasoning that we have is deductive reasoning, and it's going to flow in the opposite direction. So deductive reasoning uses general premises to make specific predictions. In other words, right, like if generally speaking, I think that all things are made of cells, I can make the specific prediction that if a dog is alive, a dog will be made up of cells. And again, same thing, if I take a big umbrella, general premise of all things are alive, I can use deductive reasoning to make a specific prediction, right? If plants are alive, plants will be made up of cells. So this kind of flip-flops inductive reasoning on its head. If inductive reasoning was going from lots of specific examples to a general big picture idea, deductive reasoning is going to do the opposite. It's going to take a big, general big picture idea and make a very specific concrete prediction about a particular thing. Okay? We use these two opposite types of reasoning to come up with a hypothesis and a testable prediction. And here's how this works. Okay. So once we make all of those observations about the world around us and we come up with a question, a lot of times we'll want to have a proposed explanation or a best guess to explain that observation or to answer that problem or answer that question that we came up with. That proposed explanation or that best educated guess is something called a hypothesis, and it's a tentative answer to a well-framed scientific question. We usually make a hypothesis using inductive reasoning. So in other words, to get from observations to hypothesis, we will have used that inductive reasoning of, hey, we notice lots of different things, specific things in the world around us. Using inductive reasoning, we come up with this idea or hypothesis for, in general, we think how this is the world works. Okay? So we use inductive reasoning to go from observation to hypothesis. Now, once we come up with that hypothesis, right, it's great to have a tentative answer to a question that we want to ask. But we are scientists, and we depend on data, and we depend on rigorous experimentation. So using that hypothesis as a starting point, we need to come up with a testable prediction in order to frame and build the experiment that we're going to use to support or disprove that hypothesis. Okay? That testable prediction is created with deductive reasoning. Okay? So deductive reasoning allows us to go from that generalized hypothesis to a very specific test that we want to perform. Okay? Once we have those two things, we can then design an experiment with variables and with conditions and control groups, and through the process of scientific experimentation, we can come up with data and hopefully come up with a conclusion about whether or not that hypothesis is supported or disproved. You've probably seen all of these steps of the scientific method before in kind of a linear form. The thing that I wanted to emphasize in Bio 111 as a college level course is that in reality, this scientific method, the scientific process, is really quite flexible. So instead of a linear chain of stuff, science kind of looks like this, right? At the heart of it is going to be observation and forming and testing hypotheses with experimentation. Okay? Now, from this, we can do experiments, and what we can do is then, share our data with the community and get feedback on our data and use that data to revise our hypothesis and do other experiments. Once we revise our hypotheses, we can go back to experimentation, exploration, and discovery and perform more experiments, get more data, get more information, and learn, do more research to find out more about the natural world. And the other thing that we can do as a result of experimentation and finding out more information about the natural world is we can share the information with society, and society can benefit from that information. Say if I learn that there's a chemical that kills off cancer cells, maybe that can be used by society to come up with a new treatment or therapy for cancer. Maybe if I learn something new about the natural world, like maybe if I learn about the presence of life on other planets, I can share that knowledge with society so we can learn more about the conditions for life, not just here on Earth, but also in other places. And we could think about how to make life on Earth more habitable for us in the long run. So as opposed to being kind of like a linear chain of events, the scientific method is actually quite flexible and cyclical and repeatable, so that oftentimes scientific experimentation will lead to more questions, lead to more discovery, and lead to more inventions for the world around us. Now, that's kind of generally the background of scientific method. What I want to do now is share more specifically the format of the scientific method that's going to come up in your tests. So if you ever see on the test a problem-solving question, right, and you will, because there's going to be at least one problem-solving question for every test, this is going to be the format of answering those problem-solving questions that I'm going to ask everyone to be able to put onto the test. So on a test, when I'm looking for designing an experiment using the scientific method, this is the kind of the format that I'll ask you guys to use. These are going to be the steps of the scientific method, and this will be presented in a linear format. And the only reason I'm asking you guys please to use this format is just it helps me find the information faster when I'm grading, and it's easier just to grade everybody's work. So of course we're going to start out with defining a problem. Okay. The second thing that we're going to do is we're going to come up with a hypothesis, a general tentative explanation for or solution for the problem or question that we're trying to ask. This must be in the form of a statement. Okay. Next, we'll have a testable prediction. This is a specific prediction that comes out of the hypothesis that we're going to use to create an experiment to find out more about that problem or to solve that problem that we're looking at. This testable prediction, again, must be a statement. Okay. The thing about scientific experimentation is that before we do the experiment, we have to make a prediction about how we think the experiment is going to go. And we do that in order to be rigorous and to be purposeful and intentional when we design experiments. And that helps our experiments be valid. Once we have that testable prediction, we're going to come up with a set of 3 variables to design our experiment. Okay? And a lot of times we'll think about these testable prediction and these variables in terms of a cause and effect. Okay? An independent variable is something that we're going to test as being the cause of a particular change. Okay? So this is something that I decide as an experimentalist. Say, for example, I'm really interested in drugs that cure cancer. The independent variable that I'd be looking at is going to be like the drug that I'm testing, or the drug that I think is going to cause a decrease in the cancer. If I'm interested maybe in, different physical therapy treatments to increase healing of a sprained ankle, right? I would be testing whether or not the different physical therapy treatments would be causing, right, healing of that sprained ankle to go better. Again, the independent variable is something that the scientist decides or tests or think is the cause of, right, something to occur. Whenever we have that independent variable, we need to make sure that we have 2 groups, a control group that does not receive that tested condition, and an experimental group that does receive that tested condition. So if we're talking about that, cancer drug therapy, one control group would need to not get the cancer drug therapy, and the other experimental group would need to get that cancer drug therapy. If we're talking about that physical therapy experiment, the control group would not receive the physical therapy, and the experimental group would receive the physical therapy. So in other words, between the control group and the experimental group, the only difference should be the presence or absence of that independent variable, the thing that's tested, or the thing that we think is the cause of an effect. Now, our second variable is going to be the dependent variable. This is the variable that we're going to measure as an outcome. It's going to be the variable that we measure as an effect. So again, if we're talking about cancer and drug therapies, right, if we think that the drug is going to cause a decrease in tumor size, right, that we think that the patients are going to be affected by the drug such that their tumor shrinks, right, the dependent variable is going to be the variable that well, it's going to be the shrinkage of the tumor or the disappearance of the tumor. We think it's going to be an effect that we're going to measure. So, if I were the clinician, I'd be measuring the size of the tumor and seeing if it shrinks. If I was the physical therapist, I would be looking at whether or not the time for the healing of that sprained ankle would be different. So I would be measuring the effect of that physical therapy treatment on that sprained ankle to see if it's shorter or longer in time. Again, if we think cause and effect, if we think testing and measuring, we as a scientist, our hypothesis would be that we think the independent variable causes the effect on the dependent variable. Or, right, we're testing whether or not the independent variable causes a measurable difference in the dependent variable. Okay. The last type of variable that we have is going to be a standardized variable, and standardized variables are just the environmental conditions of the experiment, right? So if we're talking about like cancer patients and drug therapy, we'd be talking about maybe the age of the patients or the sex of the patients, maybe the stage of the cancer of the patients. Maybe we were talking about, other medications of the patients, right? All of the things that kind of surround our experiment, right, that could affect our outcome, but we're not testing those variables. Those variables would be the standardized variables, and they're called standardized because we try to make sure that we standardize them or keep them the same between the control and the experimental groups. And that's so when we do this experiment, at the end, if there is a difference in that dependent variable, if those standardized variables are the same between the control group and the experimental group, right, that would ensure that the only cause of our effect in that dependent variable should be due to the independent variable. Okay, so again, those standardized variables, they should be like the environmental conditions of that experiment, and they should be held the same or held constant between the control and the experimental groups. Okay, finally, once we do the experiment, we should get data that we analyze. And again, those data are going to be either quantitative or qualitative. And the last step in our experiment is that we would reflect on the data and determine whether or not the data would either support or disprove that hypothesis. Okay, so in terms of experiments, a lot of times the experiments will kind of form a closed circle of logic, right? Where if you go through the steps of the scientific method, at the end, that conclusion, we're going to need to refer back to the hypothesis and kind of state whether or not our data supports or disproves that initial tentative explanation. So for a concrete example, right, we can use the problem of, does fertilizer make plants grow? That's our general problem. That's our general question. We see lots of different observations of, if we put different types of nutrients on plants, plants seem to grow bigger. If we want to formally start to do research on this, we then define that problem as, does fertilizer make plants grow better? Our general hypothesis, which is again a statement, is that fertilizer makes plants grow better. Again, that has to be a statement, and it's general. Our testable prediction upon which our experiment is made is this: 1 table of fertilizer daily for 1 month will increase plant growth by 50%. Okay, do you see how the hypothesis is generally, in general we think plants grow better because of fertilizer, and the testable prediction really kind of puts some concrete numbers and conditions to test that hypothesis. 1 table of fertilizer daily for a month, plant growth 50%. Okay. Once we have that testable prediction, we can then determine our variables. Okay, so for our experiment, we're testing if fertilizer causes the effect of better plant growth. So take a second to think about which is the cause and which is the effect, what we're testing and what we'll be measuring. Okay, and if we think about it that way, the fertilizer is what we're testing as an independent variable because we think it causes the effect of plant growth that we're measuring. So in this example, our independent variable is fertilizer and our dependent variable is plant growth. Okay. Again, to make sure that this effect on plant growth is only due to fertilizer, okay, we need two things. We need control group and experimental group, where one group receives the test, the fertilizer, and we have another group, the control group, that does not receive the test or does not receive fertilizer. And the second thing that we need to make sure that the effect that we see is only due to fertilizer, our independent variable, we need to have standardized variables be set the same between those two groups. Again, these standardized variables are environmental conditions that could affect our outcome, but because we're not interested, we're not testing these variables, we will actually want to keep them the same between those two groups. So in this particular case, you can think about environmental conditions that can affect plant growth, right? Things like the amount of water, the amount of sunlight, the temperature of the plants, the species of the plant, maybe even the initial stage of growth of the plant, okay? All of these things could influence our results. But because we're not testing any of these, they're going to be standardized variables that we keep the same between the control and the experimental groups. Finally, once we do the experiment, we should get data and analysis. Right? We should get data that we can analyze. Quantitative data might be that the plants grew 2 times as tall as plants that did not receive fertilizer, or maybe they had 4 times as many leaves. Qualitative data, maybe we could say that the plants that received fertilizer seemed greener and more turgid or more upright. Okay. And then finally, at the end, we would look at the data and make a conclusion, right? Our data do support the hypothesis that fertilizer makes plants grow better. Okay. So although we can disprove a hypothesis if the data, seem to go against our initial hypothesis, we can disprove our hypothesis. But in terms of the opposite, can we ever say that we've proved a hypothesis to be true? Well, in science, we're skeptics. And really, we only trust the data as far as the next experiment comes along. So instead of saying that we have proved the hypothesis to be correct, what we can really only say is that we have supported the hypothesis. And so far, the data that we have at this point, currently at this time, the newest data, they continue to support the hypothesis. Okay. And we do that because as skeptical scientists, right, we only trust our data as much as we have done, and only up until the next experiment that could have the possibility of disproving it. Okay, so we can either say that we've disproved the hypothesis or that we've supported the hypothesis. Now, if you take a look in your case study folder, right, the case study this week is on Avandia and its role in treating diabetes. Okay. As a kind of hint or, helpful nudge, right, probably the case study on your test will look very similar to that case study that you're seeing in your Blackboard course shell. In other words, the problem-solving question on your test will look very similar to that Avandia case study in your Blackboard course shell. So take a look at the worksheet, take a look at this, and try to come up with that problem-solving experimental design for that Avandia case study. And I've started you off. Here's your problem. Does Avandia lower blood glucose in patients with type 2 diabetes? Okay. Go ahead and take a look at the video, take a look at the case study, answer the questions in the case study, and then take a look at the rest of this slide and try to see if you can fill in the rest of the elements of experimental design for the Avandia and diabetes case study. I think this is going to be very helpful for the assignment and also very helpful for our Unit 1 test. All right, that's it. Thanks for your time. I think that's it for the chapter. So we will continue next week with Chapter 2 on chemistry and atoms and molecules. If you need anything before then, shoot me an email, mfadri@waketech.edu, and I'll see you next week. Thanks. Bye.